Okay, let's dive right into this high stakes biological explainer mapping out the epic journey of a T cell. If you're gearing up for the CSIR Net Life Sciences exam, or honestly, if you're just a fellow biology nerd who really wants to grasp the absolute powerhouse of our adaptive immune system, this is an absolute must know topic. We're going to break down the incredible life cycle of a T cell, starting from its very origins all the way to its highly specialized deployment out on the cellular battlefield. So, here's our game plan for today. We'll trace this chronologically through 4 sections. First, the thymus training ground, then the anatomy of an encounter. Next, we'll hit the 3 activation signals, and we'll wrap it up with specializing for the fight. All right, let's kick things off with Section 1, the thymus training ground. Our journey actually starts way before a T cell even has its identity. Precursor cells originate in the bone marrow, travel through the bloodstream, and finally arrive at the thymus. And honestly, you can think of the thymus as this brutal, highly exclusive military academy for your immune system. Now look at this stat, 1 to 3%. That is literally it. Out of all the millions of naive precursor cells that enter the thymus to try and become T cells, only 1 to 3% actually survive the selection process. The vast majority fail and are eliminated through apoptosis before they ever even see the outside of the thymus. I mean, it's a massive 97% failure rate. But as you'll see, this absolute ruthlessness is completely essential for your survival. So, the recruits, we call them thymocytes, mature sequentially across different zones. They start off in the double negative, or DN stage. Basically, they're blank slates. No CD4, no CD8 receptors. Guided by these vital instructional cues, like notch signaling and chemokines, specifically CXCR4 and CCR7, they develop into double-positive thymocytes, expressing both CD4 and CD8. Finally, if they pass their internal exams, they commit to just one receptor, officially becoming single-positive T cells. And those exams I just mentioned, we call those positive and negative selection, and they mainly happen at the cortical-medullary junction and the cortex. First up is positive selection. The T cell has to prove it can actually recognize your body's own MHC molecules. If it can't read your biological display boards, well, it's useless and it dies. Then comes negative selection. Here, the T cell is shown self-antigens. If it reacts too strongly to your own tissue, boom, apoptosis. This creates central tolerance, ensuring the surviving 1 to 3% are ready to fight pathogens without accidentally attacking your own body. Which brings us to Section 2, anatomy of an encounter. The elite few surviving T cells graduate and migrate out to secondary lymphoid organs, like your lymph nodes. These are basically your peripheral military base camps, waiting for an invasion. And at this point, our graduating cells are split into 2 totally distinct forces. You've got your CD4 positive cells, which are your helper T cells, and they are exclusively restricted to reading MHC class II molecules. On the flip side, your CD8 positive cells are the cytotoxic T cells, entirely restricted to MHC class I molecules, perfectly specialized for 2 completely different roles. But here's the catch, and it's a big one. T cells are completely blind to free-floating threats. Viruses, bacteria just hanging out, T cells can't see them. They rely on a patrolling police officer, an antigen-presenting cell, or APC, like a dendritic cell or macrophage. These APCs are out there capturing pathogens, chopping them up, and formally serving up the antigen fragments on their MHC molecules to the T cells waiting back at the base camp. So what happens when an APC finally shows up with an antigen? That's Section 3, the 3 activation signals. A naive T cell can't just immediately leap into battle. A rogue immune response is incredibly dangerous for your body, right? So we have this highly secure 3-part authentication process to activate the T cell. This exact sequence is a heavily tested syllabus point, so definitely lock this in. Signal 1 is antigen recognition. The T cell receptor, or TCR, physically binds to the MHC peptide complex on the APC. Signal 2 is co-stimulation. The CD28 receptor on the T cell binds to B7 proteins on the APC. And finally, signal 3 is the chemical phase, where cytokines released by the APC hand over the final battle orders. Let's zoom in on signal 1 for a second, because inside the cell, it's basically a high-speed molecular relay race. The moment the TCR binds the MHC, the CD4 co-receptor recruits a tyrosine kinase called LCK. LCK immediately phosphorylates the ITAM domains on the nearby zeta chains. Now, those activated ITAMs act like a magnet, pulling in a vital protein called ZAP70. ZAP70 fires up and phosphorylates LAT, the linker for activation of T cells. From there, the activated LAT complex just explodes outward, bringing in another critical protein, SLP76. This bridges the signal out into massive intracellular cascades. We're talking the PLC gamma pathway, MAC, and the RAC pathway. These all crosstalk and dive deep into the cell's nucleus, firing up transcription factors like NFAT and AP1 to completely rewrite the cell's genetic expression for proliferation. But we have a brilliant fail-safe built in here. What if a T cell recognizes an antigen, but the APC isn't providing that secondary co-stimulation signal? Well, if signal 1 happens without signal 2, meaning CD28 doesn't bind to the APC's CD80 or 86, the T cell knows something is off. It totally shuts down into a permanently unresponsive state called anergy. It's a critical mechanism to stop T cells from accidentally attacking harmless proteins. Now, assuming everything goes perfectly and both signals hit, you gotta keep that fire burning. Hours after the initial encounter, late-stage interactions step in. The OX40 receptor on the T cell locks onto the OX40 ligand on the APC. This late interaction rescues the cell from signal decay and massively boosts cytokine production for the fight ahead. And that leads us to our final stage, Section 4, specializing for the fight. Let's see how this all culminates in that third signal, turning our T cell into a specialized combat unit. So, Signal 3 is all about polarizing cytokines. The APC evaluates the exact type of threat using its toll-like receptors. Is it a virus, a parasite, bacteria? Depending on what it senses, the APC secretes specific chemical messengers, these polarizing cytokines. They are literally customized battle orders that dictate exactly what kind of warrior the naive T helper cell will become. This table right here, absolute goldmine for exams. Let's break it down. If it's a virus, the APC secretes interleukin-12, which triggers the T-bet master regulator, creating a Th1 cell. If it's a helminth parasite, you get IL-4, triggering Gata-3 for Th2 cells. Extracellular pathogens trigger TGF-beta and IL-6, activating ROR gamma-T for Th17 cells. And for immune regulation, to just calm things down, TGF-beta activates FOXP3, creating regulatory T cells, or TREGs. The cytokine dictates the gene, and the gene dictates the warrior. Okay, identity locked in. Now the T cell needs an army. It starts pumping out interleukin-2 while simultaneously building IL-2 receptors right on its own surface. This creates an autocrine loop. It's basically signaling itself, forcing the cell to rapidly clone into a massive, highly specialized platoon that is completely ready for war against that specific pathogen. So we've just mapped this incredibly flawless machinery of T cell immunity, from that brutal Simus training to the 3-signal activation and the final specialization. But I want to leave you with one final provocative thought. Remember that 1 to 3% thymic survival rate? It's strict, yeah, but it isn't 100% perfect. If a rogue T cell with a self-reactive receptor actually manages to slip through negative selection and bypass that anergy failsafe, how exactly do those rogue escapees trigger the devastating autoimmune diseases we see today? Well, that is a massive question for our next deep dive. Keep questioning, keep learning, and I'll catch you in the next explainer.